How to Build a Living Human Being from Dust. Frank W. Abernathy, PhD, 2015. And that is my email, and that is the website for the blog relating to this presentation. Start with dust. We're being a little silly here, but here's two examples of dust. There's dead, like moon dust, and there's live, like fertile soil. So let's examine this a little further. Living dust. Living dust is much more interesting than dead dust. It contains myriad kinds of bacteria, fungi, algae, spores, and microscopic nematode worms and arthropods, bugs, among other things. To the naked eye, however, it is nothing more than dust. Living dust contains viruses. Did you know that any kind of cell, be it a bacterium, fungus, nematode, arthropod, plant, or animal, contains viruses that live inside them? This is the living dust within the living dust. However, to be fair, scientists aren't sure if viruses are really alive or not, since they must be within a host to reproduce. How do we go from dust to human? I appreciate your patience, I really do, but you'll have to bear with me for a little while. This didn't happen overnight, you know. The living dust I am talking about starts with viruses and relatives like plasmids. A plasmid is basically a naked virus that has no outer coat like protein. The outer coat of a virus allows it to leave an infected cell and be transmitted to other cells either nearby or via some kind of vector like blood transfer or an aerosol. Plasmids can't do this. They have to go along with the original cell until it divides, and then copies of it can be transferred to both daughter cells and so on. So what exactly is a virus? A virus is a piece of nucleic acid like DNA or RNA. The ones most likely to be human ancestors are DNA viruses, more specifically circular DNA viruses. Unlike linear DNA, circular DNA has no ends to it. This is quite useful in preventing cell enzymes from destroying it from the ends. Okay, what's next? Well, this is where models come in. We can build a larger DNA circle from smaller ones as shown by the diagram below. It shows two circles representing DNA. They come together and join and fuse, and then you see that the fusion is a little bit incomplete. So how do these circles fuse? For the sake of simplicity, let's use two rubber bands glued together as an example. And you can see they've been cut in B with scissors, and they separate and form a larger circle, but it has dimples in it as shown by the little arrows, so it doesn't form a complete true circle. So we really have an imperfect fusion here, and it causes these dimples which mark the location of where the fusion occurred. Of course, you can take the two rubber bands and unglue them and restore them back to their original shapes as two rubber bands. You could also trim out some of the dimpled areas without causing the circles to fall apart. However, when you do that, you lose information in the process. Check out the next slide to see what I mean. Okay, now don't panic. I'll explain this slide. The two rectangular shaped objects under figure A, they're both labeled 1 because they're identical. Those represent circular DNAs. There's no helix, so that it's a little more clear what's going on. The rungs in those ladders are the base pairing that holds the two circles, or the two strands of DNA together. So there's 4 strands of DNA, 2 on the top and 2 on the bottom. Now, when you go to B, you start to see the rungs separate and point up and down because they're getting ready to base pair with each other, both of those replicons, and they do so in C. From C, you can go to A. I'm sorry, not A, but D. And if you look, you see that where they have recombined has been deleted, and therefore information has been lost as a result. And that's why you have a one-way arrow going from C to D. When you look at E, C can also form E. You don't delete anything, you just cut a little snip in the middle of that section, and you still have all the DNA information. And you'll notice that if you open it up and form a double circle, fusion circle, like in F, that the little black rungs in F, that's called snapback DNA, because that fusion circle can reverse completely back to E, and then C and B, and then the circles can actually separate and go back to A. Okay, in that last diagram, the what is holding the circles together is base pairing, but there must be another kind of glue holding them together, especially when they completely separate and form fusion circles, because you see some dimpling effect there. And what that would be something called covalent bonds, which are much stronger than the bonds holding the base pairs together. I won't get into a lot of detail about this here, but you can read more about this within the rest of the blog. Okay, so we have DNA circles that join together. What's next? What's next is the building up of a giant circular complex of DNA that will eventually become a bacterial chromosome. It will do so using a switching mechanism in which some circles attach together while maintaining their full DNA information, while others attach together and permanently fuse by losing some internal DNA information. The latter process is called illegitimate recombination. Okay, we're going to use those processes that we've talked about earlier, those circular DNA fusions and attachments. We're going to use that to build a bacterial chromosome from viruses, or what we would call simple replicons. So when you look at A in this diagram, you see two little circles, the replicons, and the two little dark areas facing each other are the origins of replication, where DNA replication is initiated. Now those two circles are going to join, base pair at their origins, as shown in B, just like we showed earlier. They're going to fuse, as in C. There's been no information loss. You still have your both your replicons and all the origin of replication information. In D, one of those origins is used to bring in another circle or replicon, and in E, you have three of them. You continue the process with F, G, and then you go all the way down to N, and now you have 5 replicons and 5 origins of replication. No information has been lost. G can also go to H, and you continue adding more and more replicons. It makes the chromosome bigger, and you're building the chromosome. It goes from H to I, up to J, and then it goes to K. But when you go to L, you'll notice something different. Those replicon, the origins are being cut out. Not all of it, but most of it is being cut out, and it's a permanent fusion like it was in the other diagram. And you wind up with something like M. Okay, now we're getting into the eukaryotic chromosomes, which are more similar to ours than they are to bacteria, which are called prokaryotic chromosomes. Now, when you look at this diagram, like in A, you'll notice that the long rectangular shapes, there's 4 of them, and they have the 2 origins. Those are what you saw in figure M of the last slide, and the one in the center that's got it's got 5 origins. That's what you saw in figure N of the last slide, and they're going to join together at their origins and form what you see in B, which I call that a complex replicon, or a replicon cluster. Now, as we proceed through time, you'll see that you can add additional replicons to this replicon cluster, and these are called heterologous replicons, because the origins of replication that are going to join up with the origins that are on that slave replicon at the top in A When they pair up, there is a mismatch because due to mutations and changes of the old origins of replication over time, and therefore the matchup will be imprecise, and you'll wind up with something called intron loops there at these origins that match up. You can see what these intron loops would look like in C, but we're not going to go into a lot of detail about this right now. You can read more about it at the blog. There is another way that these replicon clusters can become even more complicated, and that is by fusing together. As you see in this diagram on A, it shows two origins of replication fusing together, and you see it going down into B and then C. Those two replicon clusters have fused together. If you go down to D, you add another replicon cluster, and then you go down to E, and when it fuses, you have three replicon clusters joined together by a giant circle, and that circle, which has even more origins available for more replicon clusters, we call that a proto-chromonemata, which is the beginnings of what I call subchromatids, and you can learn more about that later in the presentation. As an aside, you'll notice that there's little squiggly lines between those slave replicons that sort of break them up like little brackets. Those are just indications that those slave replicons could be any size. Okay, we're going to move away from DNA for a little bit and talk about how mitosis, cell division, and complex eukaryotes such as ourselves may have evolved by bacterial endosymbiosis, which is bacteria and ectosymbiosis. Endo meaning within, and ecto without. So if we look at figure A at the upper left hand corner, there's two kinds of bacteria there. There's a pileated bacteria. We won't go into what pileated means. It just means it's covered with certain little outcroppings. And then there's another kind of bacterium that looks kind of like a spirochete, which is a certain kind of bacterium, but it's a motile bacterium. And these two symbiotes come together. They're not symbionts yet, but they come together in B and become what they call conjugated symbiotes. Now what happens is that not only do their cell membranes fuse together, but that the DNA it comes together at their origins of replication just like they did in the other slides. And so they are now like one organism instead of a complex organism. So they have become symbionts, a well-known phenomenon in biology. And the reason that they did this is because it gave these pileated bacteria higher motility. They could move around better, and they probably have something that they offer the other bacterium in terms of the number of kinds of materials that they can digest and absorb. So there is mutual benefit from this relationship. Now if you look down at B, there's a much larger prokaryote, which I call a phagocyte. That's number 2. And it has its own little supercoiled chromosome inside of it like you would with any other bacterium. And remember how that chromosome was put together earlier. This conjugated symbiont 1 and this phagocyte 2 join together to form something like C, and they become ectosymbiotes. They attach to the outside of this phagocyte and provide it with motility, and the phagocyte provides them the ability to get even more prey that they could otherwise not benefit from when it digests its prey and then provides the nutrients to these ectosymbiotes, 1 plus 2 in C. They're not really joined together so much by their DNA. They're just kind of going along for the ride, kind of like remoras on a shark. Now when you go down to D, some of these ectosymbionts have been incorporated and inside the phagocyte and turned into something called a prey capturing organelle. That is a little device that helps these things shoot out fibers called axopodia that can reach out and capture bacteria, prey, and pull them in, making the phagocyte even more efficient at capturing prey. These bacterial prey are pulled in through something called a cytopharynx, which is a primitive kind of a mouth that you see in some protozoa. They're pulled in and digested. And then E shows it being pulled in, and F is a cross-section of the prey-capturing organelle. It's not shown here whether or not there is a connection between the prey-capturing organelle's DNA and that of the phagocyte. It's sort of a remote connection, so to speak. Now, if you look at G all the way through K, it shows how this primitive complex prokaryote divides, and it must divide in a way that all the daughter cells get copies of everything that was in the original cell, and so it shows everything replicating in H. They begin to divide in I. There's some microtubules, sort of like precursors to what the microtubules would be in during our kinds of mitosis. The prey-capturing organelle divides, sort of like centrosomes in animals, and then you wind up with K, which is the daughter cells. Now in L, at the top right-hand corner, something very unique happens here. What happens in L is that you have another bacterial endosymbiont, because those bacteria that are captured are no longer digested. They are actually incorporated into this complex endosymbiotic relationship and become part of it and replicate in tune with all the other endosymbiotes. As you can see in M, they act like they're almost like little bitty chromosomes. And so this is beginning to look more and more like mitosis. You can see them dividing and separating in N and O, and then you get the two daughter cells in P. Okay, in this slide you see that the prey-capturing organelle is now turned into a centrosome, which is found in animals during mitosis. It is a self-replicating organelle, and you'll see that the bacteria endosymbionts have lost their cell walls, which is used to protect them in an environment. They don't need them inside this thing anymore. So now they become like vesicles or bubbles inside the proto or primitive nucleus, and it shows a little line separating those two chromosomes. If you look at B, it shows them getting ready to divide. And in C, they line up and start to divide, but the chromosomes are separated from each other. They're in their own little vesicles, and they separate independent, well, not independent, but they're not connected to each other in any way. And then you go ahead and form E and then F and form the daughter cells. In G, over time, we're fast forwarding through time, those vesicles fuse, and the chromosomes are now intermingled within the same compartment. And they go ahead and divide like they did before. Okay, we fast forward through time some more, and we wind up with a nucleus, a real nucleus that you can see in living examples would be like primitive worms and primitive insects, like certain kinds of flies. And this is one giant chromosome. All the little endosymbionts have fused together to form a big chromosome. And when that chromosome separates, as it starts to do in B and then lines up in C at metaphase, what they call metaphase, you'll see that that chromosome has a number of spindle fibers attached to it. It's not just one like it would be in our chromosomes. There are a number of them. And when it separates, the daughter chromosomes separate, they come and they move as one giant stiff rod. That's called a diffuse centromere because these fibers are lined up all up and down the length of this chromosome, and then they turn into daughter cells. Now, in this kind of division during embryonic development, that chromosome will break up, and you'll lose pieces of it as it starts to form different kinds of tissues. And my hypothesis is that what it loses in some tissues is not the same information as what it loses in other tissues. Now we fast forward some more and we go to F, and we have only a single attachment site, spindle attachment site, still a giant chromosome. Now this would be modern eukaryotes such as ourselves and mammals, but we're only showing one chromosome. I mean, you would have like many chromosomes. With us it's 46. We're only going to show one chromosome here. You get the separation, and you get G, and they come together after they've replicated, and you still have this giant chromosome, but you only have one spindle attachment, as you notice. And then when it separates, they look sort of like boomerangs separating in I, and then you wind up with the daughter cells that you've got in J. Okay, this slide is the mother of all slides, and it's a way of comparing geometric to arithmetic growth, and why geometric growth may have been responsible for the formation of complex animal life back in the Cambrian explosion. So let's look at what we mean by geometric versus arithmetic or linear growth. If you look over in the upper left hand corner, you'll see something called 1 times A, and then you go straight down 1, 2, 3, 4 all the way down to 12 times A. Now we're going to call those little dots little endosymbionts, little cells. We're not going to talk about whether they're prokaryote or eukaryote. They're just cells, and they join together, and just like we've shown in the other slides, and you wind up with 12 of them, and then those 12 fuse together, eventually fuse together, and form something called 1B, that big fusion vesicle or cell that you see at the bottom of that box. Now that would be linear growth or arithmetic growth, because you're just adding one cell at a time over a large period of time. So it winds up being 12 times bigger or 12 times more complicated than it was to begin with. Now let's compare that to geometric growth, which if we start at the top with 1 times B and work our way over to the right and go work our way straight down, you see that we take a 1 times B and we add a little tiny dot to that, a 1A. And then we go down to step 2 and we have B plus 2A, and then on down 3A, 4A, 5A. And when you get to the bottom, the very bottom, step 12, you'll see that that B has added 12 A's. And then if you head over to the right, you see that what you've done is take 1B and turn it into 2 B's. And then when those 2 B's fuse, you get 1C, which is 24 A's. Okay? And that's still linear or arithmetic growth. Now if we go from step 1 and go to the right instead of down in step 1, you'll see that you can take 1B, and if you bind it with another B, you make that C in one step. And if you go to step 2, come down, if you take that 1B and combine it with 4 B's, you wind up with 2 C's that join to make a giant cell called 1 times D, or just a D. And you work your way down that way, and you get 8 B's and 16 B's, 32 B's and 64 B's. And when you get down to 64 B's in step 6, if you will look to the left, we're comparing arithmetic growth at step 6. You have 1B with 6 A's. Okay? 1B. You don't even have 2 B's yet in step 6. But over to the right, you have 64 B's. That is the power of geometric versus linear growth. And a way of thinking about that is Let's count to 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now let's go from 1 to 10 geometrically with powers of 2 like cells when they divide. You go from 1 to 2 to 4 to 8 to 16 to 32 to 64, 128, 256, and 512. That is the power in geometric growth. All right, in this slide, we're looking at something I call hierarchical endosymbiosis. Now, I say hierarchical because if you look at the top of this model, there's something called a first nucleus, and let's say that's a simple bacterium or something. 4 of those come together in a geometric pattern, and you see composite nucleus 1, and then 4 of those come together, and you get composite nucleus 2. That's 16 of those first nuclei. Then 4 of those come together to create composite nucleus 3, and then 4 of those come together and complete the nucleus 4. So that's geometric growth is occurring because of all these cells integrating into and forming this. We're looking at the nucleus here, not the whole cell, just the nucleus. All this genetic information is being taken up geometrically in this organism. Now what you see immediately below all of this, if you go from left to right, we're going to say that we apoptotic bodies, and apoptosis is a process by which cells break down in a very organized fashion. They lose the nucleus starts to break apart and form very compact bodies called apoptotic bodies, and they start to break up in the next, and it just keeps going until there's really nothing left. It's a very organized, very complicated program that is designed, I believe, to prevent cancer, because you don't want these pieces of DNA floating around being taken up by cells, adjacent cells, which would corrupt their genetic machinery. So if you look at apoptotic bodies 4, it's almost like you're reversing the symbiosis. They break into 4 sections, and then those sections will break up into the smaller sections, and then those sections break up into even smaller ones, and so forth, until these things are totally degraded. Now there's another process that may occur at the same time as apoptosis, and that's necrosis. Necrosis is random, whereas apoptosis is organized. Necrosis is generally it's the bodies, the little nuclei or subnuclei start to swell up into vesicles, and they just finally just lyse or burst. And there's no rhyme or reason to it. It's just a random process. And so if both of these processes are going on at the same time, you'll see some things that look like very discrete compartments, and then other things are just swelling up and just sort of disappearing. All right, in this slide, it's another demonstration of hierarchical endosymbiosis. You see the 2 endosymbiotes in A off to the left, and then they get incorporated into A, and then from B to C to D they finally fuse, and then the D endosymbiont joins up with another one to make an even bigger cell, an F, and they lose their, if you'll notice, these things are starting to lose their cell membranes or cell walls, and that allows them to come together and form the endosymbiont I that you see below. The reason I have this diagram in here is because you're going to see something similar to this later in the blog with regards to eukaryotic chromosome structure. Okay, in this slide we're showing 2 kinds of eukaryotic nuclei. If you look in the box in the center, you'll see one large vesicle, and when it opens up you see 9 little paddle fans all conjugated together, and they look like the paddle fan structure or the replicon structure on the right, where you have the master replicon and the 4 slave replicons around that. These things are all within these vesicles. So there's 9, you might say, endosymbionts associated with that structure at the top. Now at the bottom, the situation is a little different because you have 9 independent vesicles within that larger structure, and they have their own, they're encapsulated separate from each other, so the chances are these things are not really conjugated or connected, because they would probably separate out as 9 separate chromosomes. So you have the giant chromosome where they are conjugated at the top, and then the 9 separated compartments at the bottom. Okay, now in this slide, the human chromosome, we're looking at one hypothetical human chromosome. It's not obviously a real one, but if you look on the left-hand side, there are 12 demarcations which indicate the vesicles that we saw earlier, and there's 12 of these. Some of these are much more simple than others. Others have much more information associated with them, but they have all conjugated together to create that linear shape that people associate with these kinds of chromosomes. Now, segment 8 is considered the centromere, and it's considered, it has to be structurally strong in order to pull this thing through metaphase, so it's got a lot of amplified, probably very similar replicons that don't have any information per se. And you see that in 5. If you go over to where it says B, you have what we call 7 units, translocation units. Now, a translocation unit, this is where we get into those subchromatids I was talking about a lot earlier in the presentation. You'll see that those circles don't have a lot of information in them, and if you look between 1 and 2, there is a demarcation in that chromosome, and that's where they have joined together with a single double origin of replication. Otherwise, there's not a lot going on in terms of information. You have your telomere, your little linear telomere at the top. So when you go down to 2, there's a lot more information associated with that one than there is with number 1. And all those little beaded black structures, clover leaves, are replicon clusters that we've seen earlier throughout the slide. They've just been condensed and packaged with protein. There's 4 of them there. 2 of them are directly connected to that subchromatid in 2. The other 2 are connected to the other 2. And so the fragile sites that they're showing here are very easily broken. They're weak spots where there's only a single origin, double origin associated with that. So drugs, any kind of what they call radiomimetic drugs or radiation, can break those and cause everything associated at that point to come out. In other words, if you broke the connection between 2 and 3, 1 and 2 would go off as a massive translocation event. Now on to the right, you see 4 what they call recombination units. Now these are the things that everybody's familiar with in cell biology. And it is the distance between the chromomeres, which are these complicated packages of replicon clusters. And you see a complex package like in 3 and in 2, and you can see the shape of those vesicles over there on the left side. You have between another chromomere, complex chromomere on down in 4, and then you have this massive amplification of replicon clusters in 3, which is the centromere, which is used for mechanical movement of the entire chromosome. So at this point, we will move on to the next slide. And in this slide, you see a smaller representation of that chromosome, that human chromosome we showed in the earlier slide in figure A. And we're going to take one of the chromatids. The chromatids, there's 4 subchromatids associated with that one. And we're going to fuse all those 4 subchromatids together, and you get some structure like you see in B, with all the chromomeres still associated with it. And then those chromomeres open up like in C, and you get all these little paddle fan arrangements where you've got the master and slave replicons. They're all conjugated together. And then those paddle fans open up, they all fuse. All those replicons fuse, and you wind up with something that looks like D, and you're getting more and more origins, single origins generated. as these double origins fuse. And eventually you get into something that looks like E, which is almost a complete giant circle. Next slide. Okay, this fusion process continues, and you see figure F where some of the things are breaking off into smaller circles, and you wind up with something like in G at the bottom where you have this gigantic rectangular shaped circle that's got all these little single origins of replication. Then you have 5 smaller circles and 3 much smaller circles, all produced by the fusion of these double origins of replication. Okay, now I think we're due for a reality check. Is there any evidence for any of this? And yes, there is, and I'm going to show you some examples with photomicrographs of some of the things I discovered many years ago at Ohio State University. Next slide. Now I find this photomicrograph, I think it's around 400 magnification, stained with a fluorescent dye called acridine orange. These are mouse cells and they're falling apart. I treated them with a little bit of acid, and what you're seeing is, to me, absolutely amazing. All kinds of circular structures are coming out of these motherships, these larger elements which were probably nuclei, and you see all kinds of hierarchical sizes, different sizes of these circular elements. Absolutely amazing. After a while, all of these little circles are released, and you see just a homogeneous but still many different size ranges, but they're all pretty much just circular elements at this point. As time progresses, you see they're starting to get a bit larger, and you're starting to see fusions occurring between circular elements. You see the dimples that I showed earlier. Those 4 arrows are pointing to fusion events that are occurring, which is going to make larger and larger circles. These fusion events occur all the way down to the level of the electron microscope. If you look at this transmission electron microscope, you will see 2 arrows pointing to what look like fusion events between different circles, especially in the lower left-hand corner. What you have to keep in mind here is that this magnification is at 62,500. Now compare that with the other circles that we've been looking at, the photomicrographs at 400 magnification. Now here's another electron micrograph that, believe it or not, is 125,000 magnifications to the best of my knowledge. And it looks all the world like a replicon cluster. You've got 3, looks like slave loops at the top, and 1 or 2, possibly 3 at the bottom. And then you have your master replicon to which they are all attached. So what is this daisy-like structure? Well, it's pretty amazing. So it's something that we need to do some research on to determine what these things are. Okay, we switch back to the 400x magnifications, and we notice the circles are getting bigger and more irregular shaped. There's still a lot of little circles here, but you're getting bigger and bigger circles, which I'm assuming are fusion circles. Now in this photomicrograph, it's one giant convoluted necklace of beads, and those beads could even be smaller replicon clusters, but it's one giant continuous circle like we showed earlier in our model. So what's next? Where do we go from here? What's going on? As it stands right now, nothing of which I am aware. There has been no published research on any of this since I first worked on it back around 1989 and earlier. So why is that? Why do you not find any mention of this in the literature? That is the subject of a page tab that I call Rantings of a Mad Scientist, which you can also find in the blog. Thank you for taking the time to look at this presentation.